Hello guys, so today I'm gonna show something very interesting, and it's all about look at this case. You have a mixed growth, or you have 2 to 3 colonies, different colonies in one plate. Like this one, we are having a mixed culture here. That is, for this case, you can see here we are having a mixed growth of colonies. So we are going to discuss how we are going to separate these mixed colonies to obtain a pure culture. So for this case, there are different methods that can be used to separate these colonies from a mixed growth to a pure culture. And of course, we have different method of separating these colonies. Of course, you can do a serial diluting method. You can do a serial diluting method. You can use a selective media, that is the likes of if it is a for enterobacteria, we have the SS media, Salmonella Shigella media. You can use the clade media. That's also a selective media. And also we have different type of media, selective media. So you can use selective media as a way of separating this mixed culture. And of course, you can also use a pour plate method. That's another fine method you can use by pouring a mixed growth into a plate. And of course, by doing this, so it helps us to identify or obtain that pure culture, so that we can be able to do other tests like the gram stain. We can do the biochemical test, and also we can do the antibiotic sensitive test. So, what is the reason behind we cannot use this mixed culture? Because one, when you're doing tests like the gram stain, it can give you a wrong result. Of course, you can get a mixed gram, maybe gram positive and gram negative growing at the gram positive and gram negative at the same time. And of course, you can find that you have maybe you have a characteristic like gram positive rods and gram positive cocci. We have the gram positive rods and we have the gram negative cocci at the same time in the same slide. So, and also tests like also catalase can give you false results because it is a mix of culture. So that's why we are going to do this separation of a mixed culture to obtain a pure culture. And also, I'm going to tell you that there are different media that can use for this separation of this media, for this culture, mixed culture. The first media you can use is the McConkey. It can be used is the McConkey media. McConkey media helps you to identify if that bacteria is a gram positive and a gram negative. Either one of the bacteria might be a gram positive and the other one might be a gram positive. We have a McConkey here. And also, it can help you also identify whether that bacteria is a lactose fermenters or an unlactose fermenters. So that's why we are going to do this test whereby we want to separate the mixed culture. And therefore, we have another media like the blood agar, because blood agar is as an enrichment media. It is enriched with the sheep blood agar, the sheep blood, that is not agar, the sheep blood. So that blood is because it has the nutrient, helps, it can support the growth of any other bacteria in that plate. And also we can use a media like the nutrient agar, because the nutrient agar is just a general media whereby it can grow any kind of bacteria. It is just a general basic media. It can grow any type of bacteria. So for this case, I'm going to use the blood agar. Of course, I have McConkey here and also I'm having the blood agar, but for my case, I'm going to use this blood agar. So we are going to use our while loop here. We are going to use our while loop here to pick these colonies, because of course they are mixed up. You're going to pick random colonies in the plate. Then we are going to streak. Of course, you have different method of streaking. We have the quadrant method. We have the T-streaking method. Those are the two ideal methods that can be used to separate. That can be used to separate these mixed culture. So these two methods are very good because they dilute these bacteria and separate them from each other by streaking. So because the method like the quadrant method, it makes 4 quadrants. So in this called 4 quadrants, as we are going down to the last quadrant, it is diluting this bacteria. These are mixed culture. Of course, you obtain a separate pure colonies. So I'm going to show you how we are going to do it. So you're going to pick these colonies like this. Just pick general colonies. Of course, they are mixed. I'm just to pick random colonies, just like that. So I'm going to make my primary inoculum, which is like this one, like that. Of course, I've picked a lot of colonies here. So you will expect here to have a lot of growth. So I'm going to streak. So this is the first quadrant. So I'm using another wire loop. This is I'm using another loop because we don't want to make the loop. We don't want to make to obtain that mixed growth. So we want to make at least to separate these the colonies. So we are going to use another loop here. So I'm going to make the second quadrant. That's like this, to make a straight line like that. Then you streak. Then I'm going to use another loop to make the third quadrant. Of course, I'm going to do, you rotate 90 degrees, make another, you make streaks. Then the last quadrant, of course, we are going to make them, we're going to make it. So we are having the last quadrant here. So we are doing like this. And of course, from this point, you can see how this quadrant, they look like. That one, of course, we're going to cover a plate. For that case, tomorrow when you will come and see the result here, you will see that these bacteria have separated from each other. And for that case, you'll be able to do the gram stain from that pure culture, pure colonies after you've obtained. After this bacteria separate, you obtain the pure colonies. You are able to do the gram stain, you'll be able to do the catalase test, you'll be able to do every other biochemical test, of course. And also it will also help you to do the antibiotic sensitive test. By doing this, so you have two bacteria. So you're going to do them separately. Not like the other one that it was mixed. You are going to do them separate because at this point when you have separated these and obtain a pure culture, the colonies separate. Hence, by doing so, you are able to obtain colonies. And of course, you can go further. If they are not, you cannot obtain that colonies pure culture exactly, you can do other subculturing, but picking the colonies that have separated from each other and inoculate to another place so that at least you can get more colonies the way you want. So for my case here, I'm going to incubate this plate and wait for the result tomorrow. Thank you. Stay tuned for more like this. Make sure you like, make sure subscribe to my channel, of course, at Microbiologist254 in the YouTube, TikTok at Microbiologist254, Instagram same at Microbiologist254, on Facebook pia, I have my page there at Microbiologist254. Stay tuned for more and let's learn together. Thank you.